So our second keynote speaker for this symposium is Thomas Hartung. So Professor Hartung, I will shortly introduce you. So he is a professor and he chairs a unit, kind of a department on evidence-based toxicology at Johns Hopkins University in Baltimore. And I learned you're also director of the CAAT, the Center for Animal or Alternatives to Animal Testing, also at Johns Hopkins University. And we know each other because you're also professor in Germany at the University of Constance for Pharmacology and Toxicology. So, well, Professor Hartung has published more than 500 scientific peer-reviewed publications, and the people in the field, they might know a specific paper that was published in Nature. It's already quite a while ago, 2009. The title was Toxicology for the 21st Century. And I learned from having a short look in the program. Today we will get the next level. So because the title of your talk will be Toxicology for the 21st Century 2.0. So we are very much looking forward to your talk. Thanks for being here and enjoy.
Thank you very much, Tanja. Yeah. Thanks for bringing me here. Feels always like coming home. I'm turning 60 next year, and most of the methods we are using in toxicology were introduced when I was not yet born or in kindergarten. I think this is something to think about, yeah. Toxicology for the 21st Century is one of my most cited articles, and the key message there was we are not 70-kilogram rats, which I think was a very important condensation of a lot of the discussion. I have a lot of slides. I made them available with this QR code, which I will show at the very end again. And I also have marked a couple of papers which I lodged for those interested, so that I can talk in more general terms. But first of all, happy birthday. I'm excited that this organization has become, over the 30 years I have interacted with the predecessor organization, and now BFR, has become a driving force in the innovation, in the renovation, I would say, of toxicology. I have to say that our work is sponsored by industry, but more importantly, 3 times more money comes from philanthropic sources, which gives us a certain independence. And the research is sponsored also by the typical funding bodies competitively. And if you miss the logo of your company or have a rich uncle, don't hesitate to contact me. I also have personal conflicts of interest. I don't go through them, just take it all with a grain of salt. I'm trying to bring the rubber on the ground, and I think this is the best way, by interacting directly with people. So this is one of my early papers together with Zebet, part of the BFR. And I think this was the theme for the next 25 years, the sophisticated way to a validated alternative method, if I had only known how long it takes. I also was even paid by Zebet for part of my postdoc time, so I consider myself a little bit part of the team here. And most importantly for me, I recruited Lena Smirnova from Zebet into CAT and my life, and she greets you with our family, and it is a successful partnership also on this level. What I did with sponsoring from Zebet was one of my claims to fame. I established the pyrogen test, and the work was early on sponsored by Zebet, as acknowledged, for example, in this paper. An assay which I developed with the supervision of Albrecht Wendel, my mentor, which I then led into a validation study of similar methods. This is all history, we don't need to go through this. But importantly, it had impact, but it had impact after 25, 30 years. From roughly 160,000 rabbits used for pyrogen testing, when these assays were accepted as valid, we are now seeing that they're being phased out, and last year's news from the Pharmacopoeia was, in 4 more years now, we will finally end rabbit testing for pyrogenicity testing. Imagine, if I had known as a young postdoc, I would never have embarked on something which takes 30 years. Yeah? And many companies who try to make this a business failed miserably because of these lines. And I think this is an incredible lesson learned. Our willingness to change to methodologies which are obviously performing better is dramatically bad, and is not even implemented in other parts of the world. It is Europe which is leading the way in this case here. There is somehow the feeling that regulatory science is the art of deceleration progress. And as a physician, there's a disease, deceleration injury. Sure, we're not running against the wall, so sometimes it feels like this. It looks much more like this. In toxicological terms, it is a repeated low-dose deceleration. You cannot really grasp it. How is it possible that people don't understand? It makes no sense to stay with rabbits if you have a human test available which is quantitative and has many other advantages. And if you had only 10 years earlier, 20 years after these assays became available, introduced them, this would not only have saved 1.5 million rabbits, but roughly also 100 million euro of testing costs in Europe. Yeah? Imagine this, yeah? We are not giving technology the possibilities to actually be implemented. How can biotech companies use this as a path towards, as a business model? Yeah. And this is the same for other assays. We see how slowly any of these methodologies are being introduced. Second point, I was part of the European Commission from 2002 in the wonderful position Maurice now has, and I was very much involved in establishing REACH, developing the guidance for industry, which I was sharing with 200 experts, and I really think it's a fantastic legislation. Paragraph 1 says, one of the 3 goals of this legislation is to promote alternative methods. This was what we wanted to hear. But in practice, what we see is a strong reliance on existing methods, and I would say actually deceleration at work. If you take the read-across assessment framework, this killed essentially read-across, which was the most used alternative method in the early days of REACH. Because it's now more complicated to do a read-across than to just run the animal experiments. If you take the extended one-generation study, we struggled to get in. By introducing developmental neurotox and developmental immunotox, it consumes now exactly the same number of animals as before, but is 300,000 euro more expensive and more difficult to carry out. We see that at the moment, one of the success areas of in vitro genotoxicity assays, there's a strong emphasis of putting in vivo mutagenicity assays. And for sensitization, finally we have defined approaches. And what happens? Suddenly there's an introduction of the need for quantitative data, for potency information, which these methods cannot develop. We still accept the guinea pig assays which don't produce them, but we say, no, please now come up with in vitro methods which can do potency. Just a few examples of the difficulty to struggle to implement methods which were developed in the 90s. Yeah? These are methods which are not recent innovations. And this contrasts very strongly with what a general public wants from us. They want us to move out of animal tests. You just saw 1.4 million Europeans signed for giving more emphasis on replacing animal testing in August of this year. In America, we're a little bit behind, but we're already split half. 50% of Americans don't want animal testing. It is certainly depending on what they accept and what they don't accept, but it leads increasingly to policies, but it does not really change the practice very much. But in contrast, toxicology is in principle very big science, yeah? Not reflected in all that one chair of toxicology in Germany after the other is closed down or integrated into pharmacology does not reflect the needs for this area of safety sciences, of regulatory sciences. Because after 40 years of the Genome Project, we see that 5% of diseases are genetic, yes. 40% have an identifiable genetic factor, but 70% have an exposure aspect, which we could address. This is a science which prevents diseases. Unfortunately, we cannot say how many we have prevented because they did not harm the substances which we put out. We would need much more risk assessments. Essentially, everything should be somehow assessed, but we cannot afford with the methodologies we have. We have pioneered quality control, good laboratory practice, validation. was all developed around toxicology. And it is also the first preclinical science which embraced evidence-based approaches, evidence-based medicine. I think these are really hallmarks of success. But what we see is a very rigid system, a patchwork which has been produced by one scandal produces one patch, and each of them is very different. So it is really a problem that we have a rigid system which is essentially difficult to change, and if at all, one by one, and not as a whole. I believe very strongly that science needs occasionally scientific revolutions, and this is the way we progress. It is not a slow evolutionary process, it is a process where suddenly we change a paradigm. And in this paper from last year, we tried to assess where we are on the path, and I believe that we are really moving from crisis to revolution, and this is very much along the line of Thomas Kuhn's famous book. Because we have many reasons. We have a throughput problem, and this throughput problem is very clear. Maurice said 100,000 chemicals are relevant to Europe. This paper from 2019, I think, showed 350,000 chemicals are registered in the 19 most developed countries. An enormous number. Almost 200 million chemicals have been synthesized by chemists. Yeah? We don't know how many there are. About 1,000 come to the market with pre-marketing notification in the US per year. The costs are tremendous. You see a shopping list here. It is not possible to pay for all of the assessments of these things. One pesticide is about 20 million to assess. That would be a good level of risk assessment. So the message here is there's not only ethical reasons, there's also very many economical reasons to consider other methods which are having a higher throughput. And this is one of the articles where we summarize this. It is astonishingly one of my most downloaded articles with more than 10,000 downloads on ResearchGate alone. Yeah? The next issue is, what do we get for all the money? It's terrible. I mean, we used the ECHA registration database after we made it machine readable to assess this. We found that each animal test on average was done 3 times, but we found, looking more precisely, for the 6 most used animal tests, the classical 6-pack, which consume 57% of all animals in Europe in toxicology. We found for each of these 350 to 750 chemicals which were tested at least 3 times, sometimes incredibly often. We found 2 substances which were tested more than 95 times in rabbit eyes, just to give you a flavor of the repetition which we find. So very solid. For 2,800 chemicals, we found they were 81% reproducible. And this was inflated by the negatives, because a negative substance stays a negative, stays a negative. 69% was the average reproducibility rate for toxic chemicals. So only 2 of 3 experiments give the same result. If you as regulators can sleep well on the basis of such results, I don't know. I couldn't. And this is not better for any of the systemic toxicities where we don't have such data, but we can at least analyze mice and rats. And mice and rats correspond in about 60% of the time. So please tell me how they could predict humans any better than they predict each other. So, I'm living now 14 years in the US, and the US was trailing behind Europe in all of these new approach methods for very long. But interestingly, things have changed in this cartoon here. Yeah, go ahead, we'll catch up in the wagon train later. Moving to technology-driven approaches to the airport, that's what I have been witnessing in these 14 years. For example, these are 3 roadmaps by the FDA, by the EPA, and by the National Toxicology Program to advance non-animal approaches, or new approach methods, as ECHA likes to call them. So something is happening here. We are trying to promote this as academics by teaching, for example, our 2 classes on these developments. We put out in Coursera for free, and in only 4 years, in less 4 years, 16,000 learners have participated in these courses. So this shows you there's a need for these new technologies, and we need to spread the word. There was clearly a watershed moment in the US, which we can date quite precisely, which was 2007, when this report came out by the National Academies, Toxicity Testing for the 21st Century, a Vision and a Strategy. So we have now seen 14 years since this, which means with knowledge doubling in the life sciences every 7 years, we have about 4 times more knowledge now than we had at the time. And this called, from our point of view, for toxicity testing for the 21st century 2.0. And exactly this we have done, trying to grasp all of the disruptive technologies which have come about in these 14 years, which were not even mentioned in this report. And I was charged by the Department of Defense to bring together some co-chairs and experts for a workshop which we held finally in April, Advancing the Next Scientific Revolution in Toxicology. The report is going to come out this month. First of all, as an internet-based report by the Department of Defense. We had observers from all of the US agencies which actively participated in this. So what does it say? The basis of this is, we see a tremendous opportunity for environment. We call for a human exposome project, actually, to complement the Human Genome Project. The exposome being the entirety of exposures which are impacting on humans. Exposure science was one of the 3 pillars which we identified, because there is now high throughput and untargeted exposomics, there's remote sensing, citizen science, there's biomonitoring, all of these elements which we are underestimating in an approach which is usually hazard-driven, and then only we ask, after we have characterized the hazard, is there also relevant exposure? We believe it should be at least on par, if not preceding. Secondly, there's the new technologies. Stem cells were invented in 2006, IPC at least, which made them really broadly available. They cannot be in the 2007 report. Microphysiological systems did not really exist at the time. Artificial intelligence, deep learning, which made it really practical, was invented in 2012. So we cannot really expect it to be part of TOX21 so far. And then there's the big one, which is evidence integration. What to do with all of these forces? These are the 3 topics we are discussing. To illustrate the first one on exposure, we've created an exposome collaborative in Hopkins in 2019. This is a report, or let's say a white paper, which came out of this. We are developing this at the moment also towards a large national network of exposomic groups. And we are combining exposomics at the moment with artificial intelligence. I like to call this exposome intelligence, EI, because it is really using the technologies to interpret, but also to extrapolate to those things which we cannot measure, because we can use machine learning type of approaches here. Technologies. I want to focus on 2 we are closely involved with, which is the microphysiological systems and AI. AI is possible because of the tremendous growth in computational power. What you see on this graph here is that computational power increased 1 billion-fold in these years, from the 60s to now. 1 billion-fold. Your iPhone is 100 times more potent than what was used to bring people to the moon. The Moore's law describes this. Every 2 years, computers get twice as potent and half the price, which holds until now. But since AI was introduced, deep learning, we see that AI capacities increase twice every 3 months. That's what we see in computational increases. And this is what is driving at the moment society. This is what is enabling that Google finds your website, which you use out of billions of websites within seconds. AI is in the end making big sense of big data. Yeah? Very simple. You need big data, otherwise you don't need AI. And the data production in the world is incredible. 84% of all data in the world were produced in the last 6 years. 84%. A robust 60% increase every year. That's what we are observing consistently. And in toxicology, we start to have big data because we organize our legacy data. We have the scientific literature, sensors, robotized testing, omics technologies, high content imaging. A lot is happening. We not just count tumors or dead animals. We're doing something which is really big. This starts with pathology, and most pathologists would say toxicology is just the pathology induced by chemicals. I have a bit different view on this, but This is a science which at the moment is transforming incredibly through image analysis, high content imaging, cloud storage, standardized interpretation. This alone is big data and artificial intelligence at work. Then you have the natural language processing. If you somehow spend time on AIs, and I'm the field chief editor of Frontiers in AI and very much interested in this, this is the increase, and this is a logarithmic scale again, in natural language processing models. Already 18 months ago, GPT-3, a natural language processing program, was trained on the entire internet with 175 billion parameters. And it's talking 40 languages, and if you ask it something, you can not distinguish this from a human response, really. And in the meantime, this has increased. In 2 months, GPT-4 is coming out, and the other ones have already reached more than 570 billion parameters. These systems can read a scientific paper in the very near future, as good or better as a PhD student. But they will not read one per week, they will read 1 million in a day. Yes, they are capable of extracting data from tables already. These computer programs can translate language into images, into figures, whatever you want. And this is coming within years. I don't go through this all, why AI is different to computational toxicology as you know it. There's a lot of aspects in this which most people don't understand. For example, that continuous improvement with increased data sets is achievable through deep learning approaches. We have shown in 2018, in a very simple first implementation, how good these systems can actually work. At the time, we took 10 million structures, which we calculated at the time still with an Amazon cloud server, 2 days crunching numbers, 180 cores, did cost $5,000. Today we do it on a laptop, exactly the same things. It produced a map of the chemical universe, and this chemical universe allows us to have things close to each other which are similar, sharing functional groups, and far away if they don't share them. Tanimoto in this for the expert. And the incredible thing, for 190,000 chemicals where there was an accepted classification, 190,000, we were in 87% correct in predicting these properties based on what we knew about the chemicals other than this information and what we knew about similar compounds. And this compares to 81% of the reproducibility of the animal tests. Yeah? Recall, the very same animal tests here. And this was, for good reason, twice covered in Nature, twice covered in Science, and in 400 articles in the lay press, because it was showing that the computer outperforms animal tests. 2018. But since no validation body has touched this and was willing to validate this formally. Fortunately, there's a commercial version, and this commercial version was at least accepted already in Australia for their industrial chemicals, including cosmetics. But the story goes on. The next assessment, for a very early version of our computer program, was testing how well do we produce now human data. And this is a case of human skin sensitization. The local lymph node assay, as the best animal test in this area, predicts 74% of human skin sensitization. 2 data sets on human data, one highly correlated one and one which was more scoping type of data sets, shows 80% predictivity. Clearly better than the animal test predicts human skin sensitization. And I said this was an early version. Last month we published on how to apply this in a batch approach. We just took 4,700 chemicals which are relevant to food in the US. Identified structures. And we did run it through this program, which was the equivalent of 38,000 animal studies, which would have costed 250 million to run. And this was done in less than an hour. Just running a batch of 38,000. And we were in a mini explorational validation, 83% correct. Which shows you again pretty much the reproducibility of the animal test. And even if you don't want to use AI as a replacement, it would at least show you where to look for problems, what smells like a problem, what to put on a back burner. But this is not where the story ends. We are about to publish about exploring this now for systemic toxicities. And I was blown away that, again, the predictivity for reproductive toxicity was 82%, for carcinogenicity 75%, and even better for the information-rich in vitro tests of TOX21, estrogenic, androgenic endocrine disruption, incredibly good accuracies. Yeah? Which means we could expand from the 10,000 substances which have been tested in TOX21 or the 2,000 which have been tested in TOXCAST to the chemical universe. And each and every case we would also get a certainty associated. The EU ONTOX project, which is part of the already mentioned ASPIS cluster, is expanding this concept, this approach now to systemic toxicity of liver, kidney and the developing brain. We are proud partners to this project, and I have the privilege of leading the AI aspects of this package. We believe that this is really a very important approach to expand this. But this is not only about regulatory toxicology. I think that we come in as naysayers, as toxicologists, far too late when a product is already extremely expensive and when it is hurtful to say no. Why not use these methods earlier? A computational method can be run by a chemist before he even synthesizes the product. This is what we call green toxicology. Benign by design is the principle of green chemistry. So this is an editorial in Toxai, which is describing the idea, know early about and avoid toxic product liability. It's a very simple thing. Alex Mertens, who is leading this in my team, just wrote the first textbook in this field on green toxicology. And just to give you a bit of flavor, what can be done. This is about dichloromethane. This is the similarity space of similar compounds. This is the toxicity space. This is the chemistry similarity space. Yeah, so how similar are they? And then you can overlay this and you see exactly where you should look for alternative chemistry. Yeah, so you can use it in a constructive mode, not only in saying, no, this is a bad chemical. You can use information economics and say, is it still worth running this test? Would you actually change your assessment based on this test because you see all of the information you have around? In a similar way, making toxicology more constructive, we were working with 16 pharmaceutical companies to develop concepts of investigative drug toxicology. And this idea of risk anticipation, but also of systematically de-risking things, which is not the regulatory use of toxicology, I think is a very important one which you should also translate to chemicals. I've been for long challenging that in vitro is just everything. We have been talking about the problems of cell culture as much as we did for others, because not everything will be computational. We still need testing. There's tremendous reasons not to trust a cell culture experiment per se. And I don't want to go through this in detail, but the important part is we have game changers. Cell culture of human cells has only really become possible with human stem cells. Before, you hardly got any cells which were primary and tumor cell lines are crap, simple crap. So, microphysiological systems is technologies which we actually in 2 workshops which we held in Berlin did assess. The 2 papers are available. They are long. If you want a short version, last year in Science we managed to get a condensed version of the 2nd workshop in a 3 or 4 page paper on microphysiological systems and the prospects of using them, especially in the drug development context. As a result of this, the NIH National Center for Advancing Translational Sciences, NCATS, made a call for proposals for running an international conference series on microphysiological systems, and we were the lucky winners of a proposal. So the MPS World Summit 1 was organized in New Orleans earlier this year. My co-chairs, Don Ingber from Harvard and Susie Fitzpatrick from FDA, were critical, but 52 organizations supported us here. We had 665 participants and, quite amazingly, 65 from FDA, showing you the enormous interest by the agency, which is so critical for the advancement of methodologies, the acceptance of methodologies worldwide, because it's the pharma industry which pays in the end for these developments. And we are bringing now the World Summit Series to Berlin next year, 26th and 30th of June. And we are very happy that the BFR is a partner to these endeavors, and we hope to see you all in Berlin next June. I believe very much that it is quality which allows us to promote these technologies. It is about the quality of the cell models, the quality of reporting on it, and the quality of validation. And Maurice has touched on this already with good in vitro method practices and others. We had published in 2005 out of ECVAM Good Cell Culture Practice 1.0, and now after 8 years with 100 people and 350 stakeholder comments in the last year, we just published earlier this year Good Cell Culture Practice 2.0, which is now expanding to these microphysiological systems. So a very important quality assurance. To illustrate this a bit further, I'm interested in brain organoids mainly. These were the first brain organoids published in 2013 by Lancaster et al. We came a bit later. We were the third group to publish on these, but our spin to it was to mass produce and standardize them. So this was the front page of the Financial Times in 2016, and since then these organoids have been used by collaborators and ourselves for a variety of diseases like autism. We were the first to show that COVID-19 is infecting human neurons using these systems. We grafted them with glioblastoma. You can do a lot of research using them. We are talking about toxicology here. So the first one, autism, is probably the most interesting one of these. This is the epidemic of autism in the US. In the 70s, 1 in 10,000 children was showing autism. Now 1 in 44 is diagnosed with autism according to CDC last year. 1 in 44 children. And the obvious question, genetics do not change that fast at least, so it must be some type of exposure. So is developmental neurotoxicity involved? We have not been able really to, with these animal tests, to show this, but since 2005, CAT, my current center, ECVAM, the previous one, and the EPA, and many others have collaborated in developing with 4 international conferences, 9 workshops, DNT, and you will hear more from on this from Ellen Fritsche. A tremendous undertaking. We have published that we can use the system for DNT, for drugs, for pesticides, for flame retardants. I don't go into details, that's for the specialists, but they work. And as a consequence, the EPA is funding at the moment to make this a high throughput assay for developmental neurotoxicity. Lena Smirnova in my team already mentioned, for full disclosure, my wife is leading the brain work here. And we have all of the adverse outcome pathways present in these brain organoids. So the idea is we create a rainbow. We put reporter genes through CRISPR-Cas into them that we can follow all of these processes non-invasively and continuously in just one experiment and combine it with 3-dimensional electrophysiology. And then this allowed high-content imaging. Just to show you one picture, this is an example. This is oligodendrocytes which have the green tag, so you can follow oligodendrocytes and myelination. And you have the blue ones, which is synapse formation between these neurons. And we are working at the moment on the next 2. The second project which I would like to highlight is about gene-environment interactions. You cannot study gene-environment interactions in animals properly. But in these you can. We introduced in this work, which Lena did publish last year, we did introduce into healthy genome a risk gene for autism, heterozygous CHAD8 knockout, and showed that it is synergistic with chlorpyrifos, which was just banned in the US last year because of its liability for developmental neurotox. And then you see synergy, and you can use metabolomics to show that the same things happen in this model as this happens in these patients. And we just received a quite substantial award to work now with 18 cohort studies with our autism center in Hopkins. 175,000 children are monitored in this consortium, and we are doing the experimental side to verify possible gene-environment interactions they identify in this big cohort. So what I showed you just is a bit of flavor for microphysiological systems. These are highly standardized human 3D models developed from induced pluripotent stem cells. We have the glial cells present. I showed you oligodendrocytes, but there's also astrocytes, and we can add microglia to make this immunocompetent. We can do this from genetic backgrounds. We can modify genes, reporter genes, risk factors, repair risk genes, or whatever. we can infect them with viruses. I mentioned COVID, we have done Zika, dengue, JC virus, we're working on rabies and others, and so on. Gene-environment interaction, there's functional assays. And the most recent development is to something which we call organoid intelligence. Quite incredible program. It is trying to give input and output to this and combine it with artificial intelligence. We have been moving forward with this. We just published on a 3D EEG type of thing in Science Advances in August. And we had a workshop earlier this year to form a community on organoid intelligence. And the Baltimore Declaration towards OI with 50 experts is going to be published in January. The last part is the big challenge is on evidence integration. I see I have to speed up a bit. The challenge is how now to combine all of this information and make sense of it. We have the same problem for systematic reviews, for risk assessment, for testing strategies, some retrospective, some prospective. This is a more recent paper where we summarized again where the state of the art is of integrating testing strategies, defined approaches, YATAs, because I think this is one way of integrating things. Evidence-based toxicology, systematic reviews is the other. We started this at the time in ECVAM, had the first international conference in Italy. I have a chair for evidence-based toxicology in Hopkins since 2009, and the evidence-based toxicology collaboration is pursuing this with the EPA, the FDA, and EFSA, and the Board of Trustees. Here you see the enormous uptake of systematic reviews in the field. About 1,000 people have engaged with the evidence-based toxicology collaboration. We're just starting a journal and an independent society for this. Last aspect, most recently, we are interested in how can we integrate probabilistically, with probabilistic risk assessment, different evidence streams. We just held a workshop in Italy around this topic with participation of your institute. So let me come, this is the last slide. The problem is we have so many aspects. We have tons of chemicals, metabolites of these, and degradation products. We have not only have humans, we have the animals, we have the ecosystem. And this all requires tons of tests, more than we can actually do. And they also produce discrepancies. How do you integrate things which do not align? We see here some of the ways of reducing this from one health to one substance, one assessment here, trying to harmonize these things and make things a little bit less complicated. But I think we really need a process now where we, by increasing and completing our knowledge, can prove harm or prove safety and know for sure. And I tried to show you how different technologies are fitting into this, how exposomics and AI play a role, how evidence generation with microphysiological systems, high throughput screening and AI can do. Then the evidence integration by evidence-based toxicology, test strategies, and AI again. And this will lead us to probabilities of exposure and probabilities of hazard. And we combine them ultimately in probabilistic risk assessment. This is the big vision on how these elements come together. And with this, I would like to thank you. Citing John Maynard Keynes, an economist who said, the difficulty lies not in the new ideas, but in escaping from the old ones. Thank you very much.